For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 4, Episode 4. Our discussion with some of our longtime listeners about what they took out of NASH-TAG and where they see the industry going in general starts now.
This week on Surfing the NASH Tsunami.
As the regulatory review these molecules going forward in the coming 12 to 24 months, we all need to be engaged, to be supportive. We all got to roll our sleeves up here. It's going to be a challenge, but we're up for it, and it's going to be very rewarding.
We have to have digital solutions to educate the patient, to stay close to them as they start to implement not only drug therapy, but lifestyle modification, and ensure their compliance. There's a whole universe there of communication to the patient that can be catalyzed.
We should really understand how to use the non-invasive tests that aren't biopsy, because once drugs come onto market, you're not going to biopsy everybody in order to put them on a drug or to monitor their process.
Every week, a global community of fatty liver disease stakeholders comes together to explore the most important challenges in diagnosing, treating, and developing medications for patients with fatty liver diseases. Join hepatology researcher and key opinion leader Professor Jörn Schattenberg, pricing and forecasting guru Roger Green, and today's guests, business executives Pam Danagher, Jerry Mabary, and Caitlin Schneider, as they each discuss their reactions to key issues at NASH-TAG 2023 on Surfing the NASH Tsunami.
Hi, and welcome to Episode 4, Season 4 of Surfing the NASH Tsunami, our look at what NASH-TAG meant to some of our listeners. Not the key opinion leaders you hear from every week, but people who have to take what they heard, digest it, and translate it into strategies and directions in their day-to-day work. We're doing this episode for two reasons. The first is that listeners occasionally ask questions like, I know what I take out of listening to NASH Tsunami, but how do others process it? This episode should give you some answers to that question, at least in the context of NASH-TAG. The second reason is to introduce a thought I will come back to from time to time in future episodes. This is my shameless pitch for you to help us improve ratings for the podcast. As more companies think about advertising with Surfing NASH, sponsoring episodes, or even supporting new podcast series, they ask us about our audience size and demographics. Without going into the arcana of podcasting, episode data out there is not very good, often likened, in fact, to the wild, wild west of data. Much of it's based on downloads, but one download can often translate into multiple people listening. The problem is that no one knows how often it's cached, which is the term for holding for multiple downloads, and when it's cached, nobody knows how many listens per cache. The most comprehensive rating system today comes from Apple and relies on a closely guarded algorithm that takes into account levels of interest and engagement with the podcast instead of simple downloads. One metric it relies on that is a bit more widely known is the number and quality of reviews that a podcast gets. Right now, Surfing the NASH Tsunami averages double-digit weekly comments on LinkedIn, or less often Facebook, and people send me notes, but we get virtually no reviews. So for today, I asked 3 people who sent me notes or called me to congratulate us for our coverage to join the podcast for a series of interviews. One was able to join himself. In the other 2 cases, they sent other folks from their company. This includes one pharmaceutical company. The second interview is with an executive who spent much of his career in in-office imaging and has recently shifted into digital apps and support. And the third interview is with someone who supports an in-office imaging product. What you will hear, I think, somewhat reinforces my belief that fatty liver disease and fatty liver disease commercialization is a bit like blind people touching an elephant. Different people touching the elephant in different places can experience a dramatically different sense of what the elephant is. That said, I believe you will also hear some common themes emerging from the interviews. I'm going to let the interviews speak for themselves, though, without pre-narrative. Each is thoughtful, each is thought-provoking. So when you're done, if you like this concept or if you don't, or if you have a question for any of the respondents, please leave a comment or question as a review in your podcast app, Apple, Spotify, whatever. All those reviews will come to us and we will reply, sometimes by reading them on the podcast, sometimes by inviting you to join us to ask them, and sometimes simply by email. One way or another, we hope to use the reviews to boost ratings, stimulate more interest and discussion in NASH Tsunami, Rising Tide, and other podcasts we hope to release later this year. And with that, on to our first interview with Pam Danagher, Senior Vice President of Regulatory Affairs at Terns Pharma. So the podcast listener who we have with us now is Pam Danagher. Pam, how are you this afternoon?
Very well, thanks, Roger. Happy to be here.
Happy to have you. So Pam is in Northern California, where everyone in the States and probably everyone who follows the States knows the weather and storms have been dreadful. I am putting out her Facebook all safe alert for her. at least the friends and colleagues who watch the podcast. And we have Jörn with us this afternoon or this evening in California, in Germany. Also, Jörn, how are you this evening?
I'm fine. My day is almost done, Pam. I'm 9 hours ahead of you, so quite a spread this time, but happy to have you here. Thanks for joining.
And Jörn would like everybody to know, although you can't see this, that he has finished decorating his office, which I commented on last week being barren, put up all his New York City and U.S. prints, including a couple of his marathon finishes. So he's good for everything life has to throw at him. So, Pam, since you've not been with us before, do me a favor, take a couple of minutes and tell our audience about you, what your work is and kind of what your career is and how you got to where you are. And then when you're done, I'd love for you to share with our audience one thing that they wouldn't know or necessarily suspect about you if you didn't tell them. You're up against some tough competition. Let's see how you do on that one. Your floor.
Thanks very much, Roger. So I'm here today representing Terns Pharmaceuticals, where I lead the regulatory function. I am a basic scientist by training, a molecular biologist by training, but have spent the majority of my career in drug development in actually the regulatory arena. So early in my career, moved out of the lab and into the part of the company that helps interpret the regulatory guidances as it relates to the work that's being done and helps facilitate interactions between the company and the regulatory bodies. And it's been overall a wonderful experience and the opportunity to work across many therapeutic areas, including in my particular case, many years in infectious disease, HIV in particular, and also nephrology, where I think we'd all agree there's a lot of unmet need. Other gastroenterology indications such as inflammatory bowel disease, again, where there's a lot of unmet need, and more recently entered into liver disease and NASH. And such an exciting time to be in this area with all these new data coming to light and lots of work to be done. One thing I've learned in my career is it does take a village. And I think that's what we're seeing now, is that we'll all have to come together on the back end of these huge achievements we've seen by, recent developments with resmetirom and certainly also very encouraging data with the obeticholic acid. And so, now we roll up our sleeves and the tough work begins. So very happy to be here. I think one thing about me, folks may not know, not sure that I have anything as quite as sublime, Roger, as others may have brought to you. Maybe something as simple as actually Canadian born and became an American citizen over the course of my career, and happy to be hailed from both countries and to bring that dynamic and perspective.
So Pam, where in Canada were you raised?
In Montreal.
So in how many languages are you fluent?
It's becoming more and more only one, but it's wonderful to go back and visit and have that opportunity. And for any of your listeners who've never had a chance to visit Montreal, it's a wonderful city. So I hope everybody has a chance to experience it.
Actually, I think Quebec is a pretty fantastic province. I spent a little bit of time in Montreal and also Quebec. And my grandfather, who emigrated to the States, came from Belgium, where what he used to do every summer was go to the ocean and eat pommes frites on the boardwalks. And after he moved to the States, they used to go to the Gaspé because it was the one place in America or North America that he could do the same thing and speak French. He was pretty happy about that. Canada's an amazing country. So thank you for that. And with that, let me just icebreaker question, okay? We're, I guess, a week and a half out from NASH-TAG at this point. And I'm wondering for each of the three of us, what impression has been reinforced, something you took out of the meeting that was reinforced over the last week and a half?
So let me start and follow up the thoughts I had coming home back from NASH-TAG was, of course, dominated by the progress we're making in the field, with the positive phase 3 data, as Pam mentioned. But then being back into clinic, I realized, from an implementation point of view, we're still at the beginning of this. As Pam said, I think we have to really roll up our sleeves and educate people out there to identify those patients we might have treatment for soon, on how to identify them. And I just finished recording actually 2 hours ago an educational format, CME format we did here in Germany with somebody speaking on CED, chronic inflammatory bowel disease, GI oncology. And I think these fields are so much more advanced or have a totally different view on how to identify patients, how the patients present, the severity of the disease. So albeit the progress in the medical arena, I think we still need some work on the pathway development. And I do see important steps being made, but that's work ahead of us.
I would definitely agree. And thank you, Jörn, for mentioning some of the parallels, right, between some other disease states. And, one thing that's really notable and remarkable, I think, when you compare and contrast potentially inflammatory bowel disease to the NASH situation, it's really the symptomatic component, right? where perhaps the diagnosis does involve, imaging, other NITs. In the case of Crohn's disease, for example, biopsy could be a parallel, but also this overlay of symptoms. And I think that's where we really struggle, obviously, with the earlier phase of the spectrum of NASH, that there is this asymptomatic component. So when we look ahead to the work to be done, what's particularly challenging, looking through a regulatory lens, is how these products are to be labeled and where the risk-benefit from a regulatory science point of view is positive. This is something where NITs potentially come in, and I think we heard a lot about that at NASH-TAG. It's something that will have to be worked through.
So I guess my impression is not so different from either of those, which is that, and this is one of the questions that I sent to you, Pam, so I'm giving away at least one of my biases. I said this on the podcast already. Scott Friedman used the phrase, the straitjacket of biopsy. He was talking, I think, although Scott is such a sophisticated and complex guy, you can never be certain where the outer boundaries of his vision are. But in the context, every time he's used it, he was talking about clinical trial development and clinical trial issues. And while that might be the case, it's struck me that biopsy has straitjacketed us in so many more ways than that it's left us, as Jörn describes correctly, I think, woefully behind even the starting line in terms of how do we actually get these drugs to the right patients and educate communities in the right way as we come out of the box. That, I think, is going to be an immense challenge.
It is a challenge, but the field is prepared. I mean, that's the other thing I'm taking away, because as Pam mentioned, we have these initiatives. We're defining and developing new biomarkers. We're linking them to outcomes, so that helps us to talk to the regulators. We are educating the peers on how to identify the sickest patients. And when I talk to my colleagues now, I always highlight the pre-cirrhotic, cirrhotic patients that are at greatest risk. So these cases, we have the tools, and now we need the buy-in of the field and the education. And that takes some time, but we're ready to roll it out. And because it will be much easier, it'll be great if we have a label at one point. And as someone on this podcast said, I think if we have shiny brochures and we can actually walk to the people, it'll be much easier then.
I agree. And I think the description straitjacket is remarkably apt, right? Because it's a restraint, but it's also constraining. And it is constraining, as you said, Roger, on so many parties. And it's really right through the spectrum. So the trials themselves are a challenge. Interpretation of the data is a challenge. Generalizability of the data and information back to the patient community is a challenge. And then the regulatory process is also challenged. And then finally, even from the point of view of the patient community, understanding how to interpret these data and even access to biopsy, if that's a needed procedure, can be a challenge across a diverse socioeconomic background.
Yes, I agree. A couple of other ways that I think became clear at NASH-TAG is investors are going to keep being hung up on what payers are going to do about biopsy. Now, that, I think, speaks to a couple of fallacies. Number one, the belief that payers will do anything, anytime they can to limit use of drugs so they make more money. I think payers are more sophisticated than that, or as a medical director, a friend of mine put it, I didn't go to med school to work for an insurance company, even though that was what he wound up doing. Biopsy, to some degree, casts a pall over some of the further removed, if you will, investors certainly, and maybe some of the insurers, over how is this disease going to be managed. Because biopsy is just not a feasible solution. I think everyone who's close to this knows it, including a lot of the payers, because they would have to pay for an awful lot of biopsies, which is not a good thing. By the way, my favorite statement about the straitjacket of biopsy came from Donna Cryer, who said that she thought it was a double metaphor, in part because it was constraining and in part because it was psychotic. You would trust Donna to say that, but I want to share it because I just thought it was pretty brilliant in the moment. All right, Pam, I'd love for you to share generally your impressions of the meeting, and the meeting first, and then the coverage of the meeting, and then I sent you a list of a few questions that we might want to specifically talk about. But let's be general first, and then we can go to specific.
Maybe one of the most impactful impressions from the meeting, and it really was right from the very opening of the reception, was just a sense of buoyancy that, the meeting was opened with a champagne toast to celebrate, the achievements in the clinical work and setting the stage for us all to move forward in that spirit. And so a very nice sense of optimism. I think it's been pointed out that the top line data are one thing. Of course, as always in life, the devil's in the details. And everybody is, of course, also interested to see the more detailed data that will emerge from the study, acknowledging that it's on ongoing and the blind must be safeguarded, things that will help aid interpretation and help move other initiatives forward, things along the lines of correlation of the efficacy findings with NITs, for example, I think is one thing that's been highlighted as very important. Deeper dive into safety, all those kinds of things that will help inform us. So that spirit of optimism was borne out through the entire 2 days of NASH-TAG. So a very nice atmosphere, collaborative in spirit, and setting the stage. I also was struck by a lot of progress in the field around non-invasive tests. As always, a nice readout from the NIMBLE Consortium, from Litmus, other efforts that are being moved forward, including the NAIL-NIT effort. And I think that this is really very encouraging for all the reasons we just discussed of the constraints of biopsy.
NASH-TAG is a special conference based on intensity and interactions that are happening. So again, very energizing, and I agree it sets the stage for the rest of the year. And now we're looking forward to see it rolling out.
If you heard the interview with Donna Cryer as part of our coverage, then she actually expressed less optimism, significant optimism, but less optimism than anybody else did. And it was actually around a topic that you raised in your note to me, and then you just kind of raised on this podcast, which is simply having good top-line data on efficacy and safety, or at least meeting FDA preset standards on efficacy and safety, didn't work out so well for NASH 2 years ago, almost 3 years ago now. And so the question is, what are the steps that we as an industry, I assume Madrigal will know how to do their work, but what are the steps that we as the rest of the industry need to be taking right now to help regulatory science get a foothold and deliver messages in ways that are going to be supportive of what's going on?
I think it's a great observation and a great question, Roger. Here again, we return to sort of kind of the straitjacket image and metaphor, and, it's constraining even on the regulators themselves. We highlighted the distinction that some of their disease states, in addition to other imaging technology, for example, it's in concert with how a patient feels, functions, or survives. It's in concert with potentially symptoms, for example, clinical signs and symptoms that can be readily measured, and perhaps there's validated questionnaires for that purpose. Nothing like this exists, obviously, for non-cirrhotic NASH with fibrosis, that generally speaking, it's a reliance on, at this stage, from a regulatory science point of view, it's a reliance on biopsy. And this is constraining for the regulators as well, because it's difficult. They make available an opportunity to progress programs based on surrogate endpoints in situations where, and guidance that lays this out very thoughtfully, that there's simply too long a time course for disease, to progress to outcomes. And so therefore there's opportunity to look instead at surrogates. And there's some very prescribed criteria that need to be met in order for a surrogate to be utilized. So it's forward-looking in that regard. However, the challenge then comes that from a regulatory point of view, decisions have to be made based on something other than how a patient feels, functions, or survives. So it's constraining right from the heart of it on all parties involved. And then I think your question, Roger, if I remember rightly, is, how do we all move forward? I think it takes a village. We've highlighted a lot of the main players here. We've highlighted industry, we've highlighted the regulators, we've highlighted payers, and the clinical community, of course. As the regulatory review, both in the US and Europe and any other jurisdictions, progresses for these molecules going forward in the coming 12 to 24 months, we all need to be engaged, to be supportive. There will be most likely scientific advisories of some kind or another. There needs to be a full engagement, and there needs to be a thoughtful approach all the way through to the end to the payers, as you've highlighted, Roger. I don't think this is going to be easy. I think I opened this by saying we got to roll our sleeves up. I think we all got to roll our sleeves up here. It's going to be a challenge, but we're up for it, and it's going to be very rewarding, obviously.
Let me just ask, Pam, you have much more regulatory experience than I do. From my point of view, we have discussed, and that's some years ago, the approvable endpoint, or as a conditional approval, which is histological benefit, and we've seen that in 2 cases now. But then we have the safety profile and safety concerns in some cases, where this benefit safety ratio then becomes less attractive for the regulators, and that's why maybe the first time through we didn't get this approved. Something I didn't see factoring in is yet quality of life. I can speak from a German perspective. That would be something where the payers would be willing to spend money on, while I think the other endpoints that are surrogates are not as well faced. And that's a whole different story in Europe, maybe compared to the US. But if we have those 3 things, we have efficacy, we have safety, and we have, let's say, quality of life, that's a pretty strong package to take to the regulators. And where do you see the biggest barrier then in getting a drug actually agreeable approved.
The regulatory approval obviously is a big piece, but it's not the only piece. It's also translating that through into clinical care settings, and then it's also the payer component, right? So I think it's each one of those. They're all equally important. Now, obviously, one has to precede the others, but I think they are all equally important. I do think it's a challenge to establish risk-benefit in the absence of true clinical endpoints, where you've made a conscious decision that, well, we're going to wait for the clinical outcomes. It does become a challenge to have a really in-depth benefit risk assessment. I think that's where it's not simple. You have to consider the scope of the labeling. It's just not as straightforward when there are not clinical endpoints. I think that's a generally accepted fact.
And that's why it's so important that we keep patients in the trials, continue these fully enrolled, and bring them to an endpoint or to the end of study at one point.
Yeah. So one of the things, when I first started talking to this community, people said they thought they were going to be the next coming of the statins. And one of the points I made was that if you look at the growth trajectory of statin use, it was driven largely by outcome studies that proved clinical benefit at endpoint. So we're a ways from that. And we're going to have to do a lot of this work without that, because a lot more people need these drugs a lot more than anybody needed a statin per se when they were launched. So I think that's going to be both an opportunity and a challenge all at once around that set of issues. So Pam, a question. In the note I sent over to you about things to think about for this conversation, I mentioned really 3 things we've not touched on yet much. 2 having to do with, to me, with the straitjacket of biopsy, which is, we all talk about non-invasive tests, but if the adjective you use is non-invasive, then the contrast is biopsy. And we're going to have to start thinking about the value of these tests, not in terms of they're better than biopsy or different than biopsy, but how do we think of them in their own right? Question 1. Question 2, back at Donna's psychotic reference, the straitjacket of biopsy is very much anti-patient centric, and this is a disease of patients. So there's a question of, how do you see us becoming more patient centric? Now, you could choose either or both of those, or say pass and we'll go on to something else, but I'd be interested in your thoughts.
I'm happy to open it up here with regard to NITs in their own right. I think you actually had quite a thoughtful conversation at the end of year podcast, and I think that was yourself, Jörn, together with Ian Rowe, and kind of talking about the transition of patients from primary care to secondary care, and are they coming with a suspected diagnosis, or are they coming with a number, right, a value of an NIT that is concerning. So I think maybe we preempted a little bit of the conversation with that discussion you had at the end of your podcast.
So that's an interesting thought, Pam. When I raised it in the first place, I think my context was a little different. Not to disagree with you. I think what you say is absolutely dead on. It's just, so here's the kind of thing I was thinking of. We're going to have liquid biopsies, and then we're going to have imaging tests. Those are different. We're going to have tests that can get read out in the physician's or reported out in the physician's office, either because they're in-office imaging or because they're computations of blood work that you would have gotten otherwise, like FIB4, versus tests that have to be done specifically and then sent back. Even if LabCorp can reflex an ELF or Quest can reflex an ELF, will that be in the hands of the practitioner when the patient is there? Because one of the points our colleague Louise makes frequently is that the ability to show a patient a scan or give them a number when they're in the office, and when you can coach them on what it means, gives them something to work on. Whereas if you call them back a week later and say, hey, here's your result, that's a much different process. So those were a couple of thoughts I had in mind in terms of how are we actually going to use these things. But I'm wondering what you think the intersect is between Ian's point and that one, both of you, either of you.
Yeah, so, well, from my perspective, I think, the discussion we had is how do we see the right patients? That, of course, comes from a specialty service where you want to see a lot of the right patients and not so many of the ones you can't offer anything to or that wouldn't need your care. So then you spend money on diagnostics that you wouldn't want. So here is always the difficulty. So we need a different type of tests to perform in these different settings. That is one. You're mentioning the value of the point of care tests, Roger, which help us to counsel the patient while they're in the office. That is good. The point is none of these are currently at the point where we can use them to, judge on drug response or something we're doing. And that's the difficulties in the discussions with the regulators. We're setting the right studies up to answer the questions, which test is linked to which outcome, and if it changes, how much it has to change to consider this beneficial for the patient. But the discussions on the biomarkers and in the different arenas, diagnostic biomarkers, or predictive biomarkers, which drugs should be used, prognostic biomarkers. That's just the complexity of the field that sometimes also, I think, in the discussions is a little confusing or mixed up. I'm just thinking we're jumping the gun a little bit with wanting to implement the right biomarker for the drugs. And I think the phase 3 trials will help us to identify those markers. We'll just have to wait a little bit longer. And then, of course, the initiatives like NAIL, NIT, or some of the NIT research consortia.
Listening to you, Jörn, what came to mind was a comment you made several times recently, which is it's not clear that we need any more NITs. Now, eventually we'll get better ones, but the challenge right now isn't that we need new ones, it's we need to figure out exactly what to do with the ones that we have. And I think that statement speaks articulately to what the challenges behind the question are. So that's great, and thank you for that. So Pam, I sent you 2 other questions if you want to comment on either of them. The one about patient centricity and the one about how you see the evolution of combination therapies emerging in this market. I know that's an area where Terns has significant interest, so just curious.
Yeah, thank you. Maybe we could have a quick discussion on the patient centricity. I know, again, just looking through the regulatory science lens, obviously a patient-centric approach strongly encouraged and facilitated. I think we've seen the Global Liver Institute do a very nice patient-focused drug development session a little more than a year ago now, and was quite informative. And I do think as medications are rolled out, this would be a very useful thing to keep an eye from a patient point of view. How is it going? Are the needs being met? Is the information available? I think Jörn mentioned earlier the glossy brochures, disease awareness, things of that nature. It's something that there'll have to be continued attention paid to whether or not the end goal is really being met as these drugs become available. So I think that's a very important conversation.
That's actually a pretty complete statement, Pam. I couldn't agree with you more. If you want to go on to combination therapies, that'd be great.
Yeah, I'm happy to comment and interested to hear also Jörn's point of view. One thing that's striking, obviously, is really the heterogeneity of disease. And I think that speaks to, potential utility for a combination approach. That might be a combination right out of the chute as the starting therapy. It might be add-on, for example. We've seen that in many other therapeutic areas, beginning with one particular mechanism of action and adding on another therapy to supplement if needed, if a complete response is not observed with the initial therapy. I note also that there are different modalities. Obviously, we have potentially injectable drugs. We have other sponsors working on oral drugs. Their uptake and their utility, the modality will play in to some extent. So this is where it behooves us to make a number of treatment options available. From a combination point of view, that is, as you've mentioned, Roger, something we've been very focused on at Terns. And again, the heterogeneity of the disease, I think, really lends itself to potentially requiring more than one mechanism of action to really suppress the disease.
From the pathophysiological point of view of this disease, Pam, I think I'm very fascinated by the combination approach. And I'd like to congratulate Terns for taking that view and look. And it's multiple pathways that fit into the phenotype, begets injury becomes regeneration becomes fibrosis over time, and it's just the complexity of the disease that's easily dissectable in an animal model, but much more complex in the human situation that speaks to the heart of my clinical understanding of it, that just putting out the fire on a vehicle that has a certain momentum is still going to make the vehicle hit the wall, but the fire is out. That's not a perfect transition, but it speaks to the need to address multiple pathways and to prevent disease progression. And for that, I believe in combination therapies. Now, it's more difficult to set up in clinical trials, the multiple arms you might need, the benefit of each single compound over placebo versus the combination that has to be shown. And I think I know you have been thinking a lot about that, and Terns, how to set it up and get a good trial design for that is a challenge. But overall, I think that's very appealing. And then we're seeing of the classes that have been successful so far, the GLP-1 and the thyroid hormone receptor beta agonists. Thinking of Terns, I think you're in a good position to follow up in that dynamic and build on the data that's been generated.
So, Jörn, here's my metaphor for the day. It comes from you. You said that the vehicle hits the wall, but the fire is out. If the vehicle is the liver, then to use a very American metaphor, the good news is that the body is its own body shop, right? Which is any other vehicle, you would have to take it to a repair shop and spend thousands of dollars and lose your car for a couple of months. And what you got back might not be anything like what you started with, but with the liver, until you get fairly far down the chain, the body actually has the ability to regenerate, which is what makes this disease so special and so challenging all at the same time.
I think you phrased it in a way that all the listeners would understand it. That metaphor just came to my head, and I'm not sure it translated well into English, but...
No, actually, I think the body is its own body shop, translates just fine into English. I'm not sure how that does out of English, but in English, I think it's good. All right, so Pam, it's been fantastic having you with us today. Anything else anything else you want to share?
No, thanks for the opportunity to participate, and looking forward to 2023.
My comment and thoughts to you, Pam, in terms is congratulations again on the drug development program. I would want to, and I know you're thinking that way, also see a commitment in the biomarker development. It's going to be so important for the field, and alongside of your clinical trial program, we'll learn so much, in particular looking at combinations, which could be a different story for some of those NITs.
Yeah, I think that's true. And Jörn, frankly, the slides that Naeem showed that you and I talked with Dean Tai about, to some degree changed my optic on all of this, which is the idea that you could map out the liver and figure out where in the liver different drugs are working better. Put a whole different foundation in terms of on combination therapy. My attitude is always, if you can see it, it's a lot more real than if you can simply explain it. So I thought that was pretty cool. So Pam, thanks for joining. Thanks to Terns for doing the podcast club. And Pam was not the Terns person who sent me notes talking about how much they liked our coverage. So thanks to your colleagues who did that. I'm not saying you didn't like it. I'm just saying you weren't the one who sent the note. So thanks for that. And I look forward to having you back with us another time, because over the course of the year, we're going to be diving into this whole question of regulatory science and the question of combination therapies. I can see you having a part in both those conversations. Our second interview is with Jerry Mabary, who helped launch EchoSense FibroScan in the U.S. and is now transitioning over to Chronwell, where he will be working on digital apps that support patients and physicians. This section of the podcast is a one-on-one interview with our good friend Jerry Mabary. Those who've been listening for a while may remember Jerry because he was part of episode 9 in season 2, where EchoSense and Chronwell and Modify Health launched their program Liver Healthy. At that point in time, Jerry was with EchoSense. He's now with Chronwell, so he's still somewhere in the Liver Healthy troika, although that's not what we're going to be talking about today per se, but I expect you'll hear about Liver Healthy in episode sometime in February. That's just a spoiler alert. Good morning, Jerry. How are you today?
I am fine. Good morning. And it's a pleasure to speak on your podcast, Roger.
It's a pleasure to have you. And Jerry, last time you were tightly scripted, and this time you're unscripted. So whereas a year and a half ago, almost 2 years ago, our listeners got to hear what a fantastic radio voice you had. Now they're actually going to get to hear what a fantastic mind you have as well. Louise had planned to be with us. She had a personal emergency come up. Jörn had not planned to be with us since this is the middle of the afternoon in Mainz, and he's doing physician things, I believe, today. So how is it in Denver today?
It is a beautiful, light, snowy day in the foothills of the Rocky Mountains above Denver. I highly recommend it for all your listeners.
Okay. That statement was paid for by the Tourism Bureau of Colorado. So Jerry, first of all, thanks for the kind words about the podcast. This is really simple. What I'd love for you to do is talk a little bit, take a couple of minutes for folks who didn't hear this when we did it a couple of years ago, a little bit about your career history and how you got to where you are now and your involvement with fatty liver disease.
I would be happy to do that, Roger. My career in brief involves I'm a biologist by education. I'm a diagnostic product development guy by career. I started out in GI diagnostics roughly 30 years ago when I was an officer in a company called Sandhill Scientific, and I conceived of, patented, developed, commercialized 3 technologies for esophageal diagnosis. Your listeners who work in the GI space will recognize tests such as impedance manometry, impedance pH, mucosal impedance. Those were all my projects. Obviously, I'm no longer involved with that company. We sold it many years ago, and I transitioned over to the liver. I had the good fortune way back in 2006 to what seemed quite accidentally. I met Laurent Sandrin, the founder of EchoSense and, of course, the inventor of liver elastography. At that time, he and his partners, who were just starting EchoSense in Europe, were looking for someone to partner with to bring the FibroScan technology to the United States and all that involves. So I started working with Laurent and Celine at EchoSense back in 2006, did did the work on the clinical trials, eventually achieved an FDA clearance in April of 2013, which was no simple accomplishment given that it was truly a novel technology with no predicate. That finally led to commercialization of the product in the United States. Those of you that remember the history of that, back in that era, FibroScan and liver elastography were primarily used to qualify patients for direct-acting antivirals, which first entered the market in November 2014. We were very fortunate that happened because that's what really drove the need for non-invasive diagnostics to qualify patients for DAA treatment for Hep C. And that's where I really learned the fundamental lesson that I follow to this day, and that's that therapeutics drive diagnostic need. There was very limited access to non-invasive technologies at that time, but what really drove that was the fact that we really had to have a non-invasive way to assess liver stiffness. So that's what led me to today. Thanks, Roger.
Okay, so and then one thing our audience wouldn't know about you and might not suspect if you didn't tell them.
What they would know about me, I used to be a runner. I once got 12th place in the annual Boulder 10K, which is the largest citizen race in the United States. And I got 12th place, and there were about 40,000 people running that year. So 12th place for my age and sex. That was the, shall we say, the peak of my athletic career, as pitiful as it was.
I can't match that, and most people I know can't either. Although I was talking offline yesterday with Jörn Schattenberg about how many marathons he's completed. That was because he had the picture of the New York and Boston marathons on the back wall of his newly redecorated office.
I wish I could make such a claim.
Yeah, well, he's the first person I've known who's talked about the agony of Heartbreak Hill from a first-person perspective. So that's great. And now you are winding down at EchoSense and joining Chronwell. Just before we dive into the talking about NASH-TAG, so people understand your various perspectives, what kinds of things will you be working on at Chronwell?
Well, it'll be an extension of what I'm doing. We'll talk a little bit about this later, I'm sure. But I think the next frontier in terms of non-invasive diagnostics and moving to the fatty liver treatment era is going to be diagnostic solutions to pull together therapeutics with optimal efficacy requiring minimal provider effort. And I also believe that non-invasive diagnostics are going to become very complex as we have different drugs reach FDA approval. And that's really the core of the efforts that you'll be seeing coming from the Chronwell team.
That's great. And with that as a backdrop, let's dive into NASH-TAG a little bit. You were there in person?
I wasn't able to attend this year, but I attended virtually, which was quite convenient.
Yeah, and in fact, and then listen to all our podcasts. So we appreciate that as well. So I attended virtually as well, and you also sent me a nice note about our coverage, which we appreciated. What struck you most about NASH-TAG this year that was different from other years, besides the fact that you were virtual this year and probably were in person most other years, because I've seen you there?
Absolutely, and obviously NASH-TAG felt very, very different this year. Everything was energized. It was absolutely palpable. I think we're all very, very excited about the positive phase 3 readout, at least a partial readout that we've seen from resmetirom. I think everyone now has renewed hope that indeed within a reasonable time period we have a chance at an FDA-approved NASH drug. And that really changes everything. I just found it to be a palpable increase in the energy level around the entire space.
Yeah, I think we all felt that. Was there a particular moment or talk in the meeting that grabbed you on that front where you said, boy, this is different, this is better?
No, I can't cite a specific session because it really penetrated all the sessions, particularly encouraged by the readout that Steve Harrison gave on phase 3 readout, and also very, very interested in what Maru Ranella had to say when she gave us an overview of what we expect to see in the AASLD fatty liver practice guidelines, which I believe are imminent to be published.
They sound eminent. What about it did you find most interesting?
I am particularly encouraged with what has happened across the board with the societies in terms of clarifying the fact that we need to be testing all patients with risk factors, specific guidance on how to do it, and how to manage the patients based on the staging that comes out of that NIT sequence, which of course Maru covered in detail. And of course, this isn't just AASLD. I'm very encouraged by the consistency we see in terms of the guidance from AGA, from ACE, and soon to be AASLD. It's just very encouraging to see all of that come together and have a direction that everyone could use.
So we're in what I like to call violent agreement on that. One of the other conversations as part of this episode is the one we had yesterday with Pam Danagher, who's a senior VP for regulatory at Terns. And part of what we were talking about in that conversation and in several others recently was how interesting and challenging it will be to translate those guidelines and the new drug approvals, given how the profession and the fatty liver community has thought about the disease and treatment thereof to date. In that context, my favorite statement has been one Scott Friedman makes about biopsy as a straitjacket. Other than the obvious, how does that strike you, and what do you see as a straitjacket around biopsy?
Well, I like the term, and I intend to start using it and claim it as my own. Obviously, the biopsy straitjacket goes significantly beyond just the challenges of getting a drug through the FDA approval process, which have been discussed very, very detailed manner on your podcast. But I also believe that the biopsy straitjacket has confined us in terms of our progress, in terms of being able to qualify patients for drugs once they are approved. If we look at the accomplishment that we saw resmetirom make in terms of NASH resolution and fibrosis improvement, I think we need to appreciate this from a slightly different perspective. I believe those efficacy values that we see for NASH drugs are a function of two things: the baseline efficacy of the drug that's being used, but I also think it's a function of the qualification process we used to get the patient on that drug to begin with. And obviously, we're all confined at this point in terms of we can only qualify patients for these drugs in clinical trials using biopsy data for the most part. I hypothesize, when we move to phase 4 and we have large numbers of patients qualified for these drugs and we're doing frequent and appropriate therapeutic response monitoring with non-invasive tests, I think we're going to learn some things from a feedback loop where we look at those non-invasive tests, who's responding based on NIT, and what their NIT values were at the time we enrolled them. I hope that we can refine over time how we qualify for the patients for these drugs in a way that's superior to what we can get from biopsy alone. So that, to me, is the hope of escaping the straitjacket of biopsy.
Do me a favor, for our listeners, paint a picture of one way that might work in practice.
Well, I'm hoping once we move to phase 4 and we're doing registry trials that we can think through it and we can use a variety of NITs and capture that data that's used to qualify the patient for the drug. And then I hope we can think through, shall we say, a more frequent therapeutic response monitoring sequence using those same NITs that we use to qualify, and carefully monitor that data for signals that tell us how we might do better with refining our drug qualification metrics. I hypothesize there will be a signal there and that we can do better, especially as we add new NIT to the mix, such as genetic factors and so forth.
That all makes tremendous sense. So it's interesting. You use the phrase NITs. And one of the, I think I said this to, Amy Articolo on episode 3, maybe, that one of the senses in which I think that we're straitjacketed by biopsy is that we describe everything that's not a biopsy as a non-invasive test. And in fact, in a post-biopsy world, there are going to be other ways we need to think about tests, right? For example, liquid tests versus imaging tests. Tests where you can get immediate response in office and therefore counsel the patient as soon as the test is completed versus those where you have to wait for the result to come back. As somebody who's spent a bunch of time with in-office imaging tests, I'm wondering how you believe we will progress in terms of thinking differently and more expansively about which tests to use when and how to use them in combination in that context. Both contexts, the one you just gave and the fact that we're not going to talk about just tests and biopsy and NIT anymore.
Good question. Certainly well stated. I think the future is, and I think we can all agree on this, is the vast majority of testing will be non-biopsy, non-invasive, if I can still use the term. And I think we're going to have a wide variety of tests that need to be done at the right time in the right patient and used in the right way. And there's a lot there that we just don't know yet. Obviously, we have the wet biomarkers. We have the device-based biomarkers biomarkers such as elastography and other methods of detecting and quantifying extent of hepatic steatosis. I think there's a strong future for combined markers that combine wet biomarkers and values that we get from devices, citing things like FAST and MAST and potentially MEFib. I think genetics is going to come into this as we move to customized, personalized medicine. And I think there'll even be a role for artificial intelligence, not only as a standalone, but this is going to get so complicated when we look at a battery or a sequence of tests that there may be a role for artificial intelligence-powered techniques where we look across a variety of tests and we better analyze what those tests are saying rather than siloing our tests and considering their values as standalones.
So that's interesting because I've heard AI's value in the long term described in two other ways, but never exactly that one.
Well, I've never heard anyone else state that as well. That's me kind of reading the tea leaves. And it just seems logical to me that there may be a path there that takes us beyond siloed tests where we consider them as standalones. That to me seems very inhibiting.
And it seems to be once you familiar with the idea of a blinding flash of the obvious?
Yes.
Okay, I would call your statement a BFO because we think about AI right now in the context where we can aggregate large amounts of data, which we cannot yet do with the artists formerly known as non-invasive tests. Because even if you take all the data, as, for example, NITs is doing and as others are doing and trying to pool data from existing clinical trials, you're not going to have a large enough data set to run AI in the ways that you're talking about.
Correct. Yes.
However, once you start getting hundreds of thousands or millions of patients onto drugs and they wind up being in phase 4 registries or in the integrated medical records of electronic health records of large systems like Kaiser, we should be reasonably quickly, like in a matter of a couple of years as compared to a decade or two, able to aggregate enough data to start asking that question.
That's my perspective exactly, Roger. I think the only path to this is in phase 4 registry studies, large numbers of patients using a wide variety of tests to qualify the patient and frequent therapeutic response monitoring. I think that's the only way we get the data to stand a chance at these kinds of approaches.
I'm buying all that, except what do you mean by frequent therapeutic monitoring?
Well, we're currently constrained by insurers. For example, elastography is constrained by the majority of insurers. It can only be done once every 6 months. We have a therapeutic timeline much faster than that when we're talking about, for example, metabolic drugs that lower steatosis in the liver and improve the metabolic profile. So I think we're going to need to think about how often do we need to test in order to get the signal we're looking for. Please appreciate the history of a 6-month elastography test is it's based on the fact that fibrosis responds fairly slowly. And we may be talking about signal here that responds much more rapidly than fibrosis alone. And I think we need to think about that.
That's interesting. One of the thoughts I'm having is, and Naeem showed these slides in the meeting, and we talked about it, I think, in one of the episodes, the work that HistoIndex has done to do zonal analysis and to identify aldafermin works here, but resmetirom seems to work there, and here and there are not the same zones of the liver. So I suspect, I'm going to run with your idea for a second, that one of the challenges we will have over time is bringing that data set, which will not be part of standard practice because it's biopsy-based, together with the, probably in this case, liquid tests, to figure out how do liquid tests translate, and are there liquid tests that translate into zonal analysis? Can we put all that together? Because if you could do that, then either that or genetics will get you tailored therapy.
Yes, there's a whole universe out there where we have ideas on things that we think we should do that may yield fruit. And I just think this draws attention to the fact that we're still in the early days of knowing how to qualify patients for these tests and monitor therapeutic response. And this is going to get exponentially more complicated when we have more than one drug and we start to do combination therapies.
So now that you've led into combination therapies, talk to me a little bit about how you think that's going to work and how that's going to fit in with everything else we've been talking about.
Well, I believe it's inevitable that we will have combination therapies. I think it will be required to use combination therapies in certain circumstances. So I think there's no question that we're going to have to move to that, but it's going to be very, very complicated making decisions on what to do and when to do it and how to monitor the response. I think clearly what we're going to find is insurers are going to be interested in detecting non-responders early and getting them off the drug and potentially potentially onto another drug. You talked about in your podcast the concepts of an induction drug that quickly induces a therapeutic response, a specific target, and then perhaps the patient goes on a different drug or a different dosage. My hypothesis, my view of the future is that's precisely correct, but it's going to become very, very complicated, and we're going to have to make it simple for the average clinician who doesn't think about this stuff all day to do this correctly. And that to me is going to require some digital solutions that guide the provider through these are the things you should consider doing next, and this is how you consider the information being provided. A lot of complexity.
So one of the things I believe is that complication is the enemy, but complexity is actually the solution. That if you take a look at complexity theory is the straight line through a muck of data. If you're willing not to take the engineering perspective, which is that error is everything, and to take a perspective that goes fundamentally, there are only 6 different kinds of harbors in the world. No 2 are identical, but they all take one of 6 basic shapes. I'm thinking that your digital solution might really help with that.
Yeah, that's my view as well, and I think that's where you'll see development activity.
Okay, so that's a physician-centric solution, really. It's about making the physician's task more manageable. Now, that will have benefits for everybody in the chain, don't get me wrong. But I'm wondering, other than making it easier for the physician to talk to the patient, where is the patient-centric piece in that, do you think?
Yeah, excellent question, and an area of strong interest for me. I think there's much to be done in terms of educating the patient, informing the patient on their specific condition, motivating them to take the correct action to improve their condition, giving them positive feedback as they make progress. I think there's a lot we can do in terms of clarifying diagnostic tests so that the patient can actually understand them, appreciate where they are in the progressive disease process, and motivate them to change. I think all of us need to think about how we can better express the information we get in diagnostics to the patient and how we can better motivate that patient to take action. And of course, that's a big part of what is being advanced and evolved with lifestyle modification. We need to remember one of the therapies that we have to choose from is not just another drug. We're told by the AGA and by ACE that lifestyle modification is a baseline core therapy that should be deployed first. I hypothesize, my bias is the lifestyle modification should be, will likely be used in concert with these NASH drugs. And that means we're going to have ways that don't take a lot of provider time, that effectively express this disease in lay terms the patient can understand and be motivated by. I think we have to have digital solutions to educate the patient, to stay close to them as they start to implement not only drug therapy but lifestyle modification, and ensure their compliance. This is going to require digital tools like apps, mobile apps that are constantly communicating with the patient, remote patient monitoring on things like weight, activity level, medication compliance. I think there's a whole universe there of communication to the patient that can be catalyzed.
So, spoiler alert to our listeners, on March 1st, we're going to drop an episode that's exactly about this subject and digital therapeutics and digital apps and how they support. And I expect that either Jerry or his colleague Joe Rubenstein will be part of our session that day. So you'll get to hear Jerry talk more about this particular issue, or Joe at that point in time. I think those are all the questions that I was hoping we'd get to cover in a remarkably efficient 29 minutes, which will be less than that when we filter things down as we do. What other issues emerged from the meeting beyond these that you would like our audience to be thinking about?
That concludes my current thoughts, Roger, but I would like to conclude by making a few comments about the Surfing podcast and what it means to me, not just for NASH-TAG, but ever since you began this effort, you have become the third rail of keeping me current with my knowledge, with everything that's going on in the fatty liver space. I do everything I can to read publications that are relevant to what I do as they're published. I certainly listen to webinars. I go to conferences, but without question, the clearest and most concise information stream that I have is your podcast. To listen to thought leaders who are involved in all of these developments talk about not only about what we know, but talk about what we don't know and what we need to pursue just has tremendous value to me. And I have a huge amount of gratitude for you and the team and what you do. And I'd just like to publicly say thank you. for what you do.
Well, so first of all, thank you. Second of all, those who know me know I'm blushing right now, although you can't hear that in my voice. And you also should know that Jerry did not have to say anything about this podcast in order to get into this discussion. So thank you so much. Where it leads me is that one of the things I will be talking about, either in the introduction to this entire episode or at the end of it, is that we're asking our listeners who feel the way Jerry does to go on whoever. Jerry, how do you listen to this podcast? Who do you get it through? Do you go directly to Buzzsprout or do you go through like Apple or someone like that?
I use Apple Podcast app.
So what we're asking is that everybody who uses a podcast app write reviews for episodes that you like and talk about them. The reason we ask for that is that reviews actually drive ratings, and they drive listeners, and they drive ratings, and they drive consistency. And the ratings drive greater presence, and that will enable us to get more information out to you faster and for you to find it easier and more currently. So thank you for the kind words, Jerry. The only thing I'm going to ask you to do is put it in your Apple review of, not this episode because you're talking, if you want to put it in the review of this episode, gosh, they were so smart to have that guy Mabary on as a guest. Go for it, but when you can, all right? Beyond that, let me just say thank you for being with us today. I've known Jerry since NASH-TAG 2020, I want to say. I started talking with you at that point in time, and you, my friend, have been a tremendous source of insight for me in terms of understanding where things are going in the device space and how to see opportunities. So one thank you deserves another. I'm excited to see you transition to Chronwell. Thank you for all the guidance you've given me over the years. And with that, let's wrap this conversation. I'll move on to the third and then to the end of the episode. Have a great day, my friend, and we'll see you on the podcast, you or Joe on the podcast next month.
My pleasure. Thank you, Roger.
Bye-bye now. Our final interview is with Caitlin Schneider, the clinical development lead at Sonic Incytes. And our third interview for this episode is with longtime friend Caitlin Schneider, the clinical development lead at Sonic Incytes. Hey, Caitlin, how are you today?
I'm doing great, Roger. It's good to talk to you again.
I haven't seen Caitlin since CLDF last year in Scottsdale, where we talked quite a bit. I think we've been in a couple of phone conversations since then, but that's probably about it. And it's been an exciting year for Sonic, huh?
It has been. We've been doing a lot of work and gathering a lot of data, and it's great to be able to go to all these different conferences and meet everybody and show them what we've been doing. We've got our latest clinical study is reading out right now, so we're really excited to be able to show the comparisons between Velcure and MR imaging.
That's fantastic. Happy to hear that and can't wait to see it published, and maybe we'll figure out how to talk about it on the podcast at that point in time. For today, do me a favor, since you're not new to me, but you are new to our listeners, please take a couple of minutes and just tell them about yourself and your history and what you're doing now and how you got to this place, and then follow that up with one fact about you that people wouldn't know if you didn't tell them. Something a little different or off.
Okay. So I am a biomedical engineer by training, and I've been working in ultrasound and ultrasound elastography for almost 20 years now. I've started off working in surgical robotics, so working with ultrasound elastography, trying to give surgeons back their sense of touch through ultrasound. And then I also worked in fibrosis development with kidneys and kidney transplant. And as I was doing my graduate work at UBC, my supervisors started the company Sonic Incytes, and I knew that it would be an amazing opportunity for me and also to see our work and our research out in the hands of real people and actually making a difference to patients and providers. So I've always been an engineer with a very clinical focus, and so working in clinical research with Sonic Incytes was a great fit for me. And I've been working here for the last 6 years.
So I should mention that, just real quickly, this is my Caitlin news. The last trip I took before the pandemic in March of 2020 was to bring Caitlin and her then CEO down to San Antonio to meet Stephen Harrison.
That was a very interesting trip, and I remember we made it back a day before the quarantine. So basically the day we flew back into Vancouver, if we'd been a day later, we would have been stuck in quarantine for 2 weeks. So that was good timing.
Timing is everything. Okay, so one thing besides that our audience might not know about you if you didn't tell them.
I know that there's a bit of a musical theme with these, so I will stick with that. And one thing that people probably wouldn't know about me is that I played tuba all through high school and college. So I was in marching band as well.
Do you know what makes that so funny?
What?
You are the second person whose story was I played tuba through high school.
Oh, really?
Do you know who the first was?
It's a great instrument. It's the best. It's solid base of a band or an orchestra.
Do you know who the first was?
I don't.
Me.
It was really? That I didn't
really.
know.
I played in the all-county band in high school and sousaphone. And you're right. And I have the only singing voice I have that survived the 20s is a baritone. And I can hear underpart harmonies on everything anybody sings because of the tuba sousaphone training.
Yeah, that's all I hear. All I hear is bass lines. It's great.
Well, you were raised in the U.S., right? So Canada wouldn't work for you on this one. But But the Star Spangled Banner has this great bass line in the most classic rendition through the middle of it and onto the back. Every time I hear the Star Spangled Banner, all I hear is the bass line for the last stanza.
Interestingly, I don't think I've ever actually played the Star Spangled Banner, but I will have to go back and listen to it now.
My pitch is not good enough for me to embarrass myself in front of all our listeners by humming it to you, but maybe I'll do that after we get off the call, after we get off the recording. That's really an interesting place to start, Caitlin. Thank you. I feel this is a very connected interview already, if only because we'll hit all the same bass notes. So you commented on, you said you really like going to all the conferences and hearing presentations and sharing data. What was the one moment or thing that you took out of NASH-TAG 2023 that you'd like to express to people before we start talking about specific questions and answers and things?
This was my first time being able to go to NASH-TAG just because of COVID and travel. So it was great to be in such a cozy location and be able to talk with some of the people there. And I think that, being able to see the presentations of the different data that actually is now finally positive, I think that was my biggest takeaway, that people are really looking forward to having some pharmaceutical options on the market. So looking at the Madrigal data as well as OCA, and then also some of the positive phase 2 studies that have been shown. So just having that as a starting point for where the field is going is really exciting for us.
Yeah, I think that's right. I mean, for those who don't know, Velacur is primarily a device for in-office use, which means the more people are treating in office, the larger your potential market is. So I think that's not the only reason it's a good thing for you, but certainly one of them. So why don't we stay a little bit in testing? Okay. Probably my favorite quote of the last year, which I've been using a bunch recently, and Scott Friedman and I both used it in covering this conference, was his description of biopsy as a straitjacket. And I wonder if you would comment from your perspective on some of the ways in which biopsy is straitjacketing the entire fatty liver space right now, and things that we would like to be able to shed over time.
I totally agree. And I think that that's a good comparison or metaphor for how it feels. I mean, for us in particular, being a non-invasive test or a test for both liver fibrosis and steatosis, having to compare everything to biopsy becomes very difficult. For us, we always have to go back to that gold standard, but because of its limitations and the fact that nobody's doing it in clinical practice, for good reason, it makes it very difficult for small companies to have new improvements on tests, but still always have to go back to this invasive and procedure. So what we've been trying to do to sort of free ourselves from this is always using MR-based imaging. So looking at MR elastography and MRI PDFF as our gold standards for comparison.
So question for you. When you talk about the unreliability of biopsy, which I've taken to calling the pyrite standard because I think at best it's fool's gold, which one creates a bigger challenge for you? Simply getting results that you can compare to Velacur in the same patients, or the rather exceptional level of variability around biopsy results?
Our biggest concern for comparison against biopsy has been just getting the biopsies to compare to. So that is always our first challenge. And then a second challenge would then be, of course, the variability amongst reads and how to do that, especially when you're trying to gather a few biopsies from one study and a few biopsies from another study. We're going to compound that variability in reading and also compound the variability in timing of when the biopsies were taken and all of that. So for us, it's a huge challenge, and it's just really not a good way for us to move forward as using these biopsies as a pyrite standard.
You say that as a result, you've taken to comparing to MRE and MRI PDFF more frequently. Are there situations where choosing not to compare to biopsy hamstrings you in any way, shape, or form? Or
a little bit. So even when we've been talking with different providers and other opinion leaders in the field, even they still want to see some amount of biopsy comparison. And so we are still tied, no matter how much we try to get away from it, still tied to needing some amount of biopsy data, no matter how hard we've tried to get away from it.
One of my favorite moments at NASH-TAG, and it actually was a very, very small moment, almost a flickering moment, although we had a conversation with HistoIndex about this, was Naeem El-Khoury putting up the HistoIndex slides that showed that with second generation harmonic convergence, you could actually start to look at where drugs worked in different parts of the liver. Now, I know that for an in-office device, you have better depth and granularity than the other available other currently marketed devices, at least. Does the idea that that can be done provide you with any benefit or anything of value? Or do you think that's something that's simply going to be idiosyncratic to AI and second generation or other forms of biopsy reads?
So yeah, we are able to measure quite a bit more of the liver, but we are always averaging over that whole liver. So we're looking at kind of how the liver as a whole is reacting to either a therapeutic or disease progression or regression. So having, I think having the granularity that something like histoindex would provide is, I think, a great step forward for the just general field of understanding how the therapeutics are working and what might be going on inside the liver. And then in-office techniques such as ours can be used just to see, the overarching trend in a patient's change over time.
That makes sense. I mean, Naeem's comment, I think, in that presentation was that biopsy is dead in the clinic and alive in the lab. So that would be, I think, a pretty good manifestation of that. Any other ways in which biopsy affects Sonic's ability to communicate, benefit, or anything else? Because the straitjacket is different people in different ways. I'm wondering if there are any harnesses that we're missing here.
It's hard, this constant reliance on biopsy as the only surrogate outcome from like a regulatory standpoint is something that just really needs to be moved past. And I know a lot of people are talking about it, but it definitely does straitjacket us. We really need to have more clarity from both the regulators and the payers about what the future is going to hold. Like, I know that a lot of people say, oh, we shouldn't talk about this until we have a drug approved, but I think that that's too late. So we should really understand how to use the non-invasive tests that aren't biopsy, because once drugs come onto market, you're not going to biopsy everybody in order to put them on a drug or to monitor their process. So having that clarity, I think, is actually key to a lot of the testing that we're going to be doing and moving forward in general.
When I think of the straitjacket of biopsy, what I think of, in fact, is exactly what you just alluded to, which is we can't conceptualize how patients are actually going to get treated and tested and evaluated. Because on the one hand, we've got these guidelines that are idealistic, but it's going to take a long while to get there because lots of people in lots of different settings have to be educated. And on the other hand, we have today where everybody's frozen around biopsy. So I believe it's limited our ability to envision the nature and scope of some of the challenges we're going to face, and as a result, imagine or create solutions. I believe your answer kind of works on all that, so I'm good with it. If there's anything else you want to add about biopsy, go ahead. If not, we'll move on to next question.
I guess it's just to kind of keep going in that vein of trying to figure out how to move away. Like, I think it's very important for the community as a whole, including, pharma and biotech, to help support some of these different options by including them into these biopsy clinical trials. I know that there is a lot of work being done with ultrasound-based markers and blood-based markers, but I think that needs to continue, and I think that they need to maybe realize how important they are to the future of some of these tests. The only way that we're going to get past the biopsy is by proving out that something else works just as well and can predict, patient outcomes with better, maybe even better accuracy than the biopsies.
I would bet on better, frankly. Looking out in the future at some point, I would bet on better being the right answer to that, if only because there's enough variability around biopsy as commonly used.
Yeah, there's, a huge amount of, and you look at the amount that you change from baseline depending on who reads, and that's tanked a few clinical studies already. So getting rid of that variability through the use of, maybe not just biopsy, but a biopsy in combination with other tests that are being done. Just the sole reliance on biopsy is somewhat straitjacketing for sure.
We're going to circle back to that on question number 3, but let's move on to question number 2. I want to thank you, first of all, for using the phrase non-invasive testing and the first time correcting yourself to tests, because I'm assuming that's because you listen to the podcast, because you smiled when you did it. If you did, then that's become kind of a big thing of mine. By the way, that's part of the straitjacket of biopsy, right? Is the adjective we're now using for tests basically mean not a biopsy?
Exactly. That's exactly what I was going to say, is that this term non-invasive tests is actually referring to everything else other than biopsy. So really, it should be just biopsy and tests.
So I consider you one of the more thoughtful people I deal with, and you take time to answer questions on lots of subjects, and you're living in this zone. When you take away the word non-invasive and you then have to start segmenting tests, the rest of the tests, in your your mind, how do you differentiate them?
So I would differentiate them into sort of two, either blood-based tests and then imaging-based tests. So the blood-based tests kind of tell you what's going on as far as inside the patient and how the patient's reacting to that disease state. And then the imaging tests kind of give you an outside view of it. So it's a holistic view of, is there stiffness? Is there fat? How is the tissue itself reacting to the liver disease? So those would be my two, I think, broad categories.
Okay, so that would be my first cut. I'll share with you my second cut, because in some ways, I think when you deal with actual issues of patient management, they might matter as much. There are tests where I can immediately give the patient feedback, either as a number that I can compute on the fly, or as a picture I can show them, as compared to things that someone else has to analyze and the patient isn't going to get the results for a while.
Also very true. So having the results that come back immediately that you can talk about with the patient in the office, I think is very motivating to patients. I feel like they are much more engaged in that way than if they were just given a number on a sheet of paper 2 months later. And I also find that people really enjoy looking at the images themselves. So having that visual feedback of what your liver looks like, it's not something that a lot of people get to experience. And I think that that is something that they actually quite enjoy. And when I'm working with patients, I do like to show them the features of their liver and have them be able to see it on the screen as we're doing the scans. They really enjoy that quite a bit.
Do you have experiences where you run those tests on people whose livers are in pretty bad shape?
Of course we do, yep.
Well, do they enjoy it as much as the people having had a Vellicure and a FibroScan and an endoscopic test and having a liver that's in pretty good shape? I enjoy it, but it's confirming that I'm okay. When you do that with people who aren't okay, how do they react?
I think it depends a little bit on where they are in their own journey and how they feel about what they're doing in order to maybe get better. They still, for people who perhaps are working hard to improve their liver health, then being able to see either their numbers stay the same or generally trend better is a huge thing for them. And I think that it helps quite a bit. So as long as they know what they're expecting, I think it's okay. We have had a few unexpected results, which can be definitely a little bit of a shock to people. But I think just having them know what they now need to do is also important.
Yeah, I guess that's right. I'm remembering my own experience in San Antonio, if you recall, where we tried a different device where the first person trying to run the test couldn't work around my rib cage. And I got to watch a whole bunch of people turn green at what they thought they were seeing. And then it turned out that wasn't what they were seeing. And then it turned out we did a Vellicure and that confirmed that it wasn't what I was seeing. But so I've actually had both experiences. I hadn't even thought of that until you started talking about people who get unexpected news. But I'm remembering that moment of watching two people turn green, one saying, has anyone ever mentioned the idea of liver transplant to you? And realizing that they weren't kidding. That was not my favorite moment.
Yeah, so I think having the reliability of these tests is exactly that. It's very important because you don't want to give somebody results that are not true. So having them be validated in a lot of different contexts and a lot of different patients is something that needs to be done and proven out before they get used in a general context.
So I guess one more question I've got for you, which is another theme that I thought came out of the conference, is the whole idea of how do you focus on the patient, or what the marketers will call patient centricity. What do you see as we start to do a better job of thinking differently about different tests? Do you see that benefiting the patient? Do you see focus in ways on tests in any way that's going to make it easier for the patient or better for the patient?
I mean, I think all these tests that are not biopsy are obviously going to benefit the patients. And I think having more than just a single test option for patients is also going to be beneficial. Not every test is going to work for every patient, and not every test is perfect. So we need to make sure that we have, a good arsenal of tests that work in different contexts as well. So tests that work better in a rural setting, for instance, tests that are more portable, tests that can be done in the back of a van like they're doing in the UK now, which is great, being able to travel to the patient. So making sure that all the patients have the opportunity to be screened and to be tested is the most important thing.
So actually, when I asked you how to differentiate, things you can put on the back of a truck might actually be a form of differentiation.
There's been a focus in clinical trials as well to be more decentralized, to make it easier for patients to be enrolled and screened for clinical trials. So I think this is all kind of related to trying to focus on what is most beneficial to the patients and how to get them more engaged and more involved in the whole process of their own healthcare as well.
I think that's everything I knew that I wanted to ask about today. When's the next time you have major data coming out?
We should have journal papers published with the complete data set That's from our last 2 clinical studies, hopefully submitted this year, published by the end of the year. So that's always good to look forward to. And then we're constantly reanalyzing the data that we've collected and improving our algorithms and improving the technology that we've built. So you can always look forward to new abstracts at EASL and at AASLD this year.
That's great. So Caitlin, thank you so much for making the time to do this. And it's great to see you even through a screen. Not sure whether I'm going to get to see LDF or not. I'm hoping I will. If I do, maybe I'll see you there.
Yeah, maybe I'll see you there. It's good to talk to you. I know it's been a while, so it's always great.
It is always great. Thanks, Caitlin.
And now for the Season 4, Episode 4 Business Report.
Slava Ukraini, Heroyam Slava, glory to Ukraine, glory to heroes. I was listening to a program today on radio where a Ukrainian advocate in the U.S. said that it is as important as military aid from the U.S. that we maintain loud support for Ukraine. He said when voices from overseas grow quieter, that's when the true atrocities and bombings like the recent Dnipro incident occur. This voice will not be quiet as long as the war continues. Slava Ukraini.
Let's talk about reviews.
I mentioned this during the podcast. One of my efforts over the past month has been to learn how to boost our ratings. I learned that ratings are scattershot and not particularly effective, but that the best rating system of all may be the Apple algorithm. That algorithm tracks activity around a podcast rather than simply checking a number of downloads. The best way for you to help us in our ratings and to demonstrate interest and activity is by filling out a rating for your episode with your podcast distributor, Apple, Spotify, whoever. That review not only tells potential listeners that this is a quality podcast, it also improves our ratings. So please, if you like us, tell your distributor. Grace comes back and says some things to update, and I continue. We will post our list of 2023 conferences within the next week. We also have 3 episodes in February covering topics we've never addressed. On February 1st, I will be on vacation, and Louise will bring you an episode from Australia about Australia. Later in the month, I will have episodes on pediatric fatty liver disease and on digital apps. The latter episode will give you your first chance to hear about Louise's new My Life 365 app.
Thanks for the strong start to 2023 listener numbers.
Download numbers do not drive ratings, but they do provide us with a relative measure of interest. Our January download numbers are back to the levels we saw through most of the second half of 2022 and included our single biggest download day ever. Not a bad start. And with that, I'm off. Thanks to Jake, Magic Mike, Eric, Steve for everything you do. Enjoy this episode. The weekend conversations will include the 3 interviews in this episode in full and some of the excellent sections of our NASH-TAG coverage we have not yet shared in conversation form. Monday, we'll be recording another exciting episode of original content, which will post Wednesday night. So until then, stay safe, surf on. We'll see you on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.